for our next question, we need to draw diagrams and label them neatly. So the first one of them is of transcription. Now we already know transcription is the production of mRNA with the help of DNA. A few things before we have a look at the diagram we need to know. DNA is double stranded, RNA is single stranded, and the enzyme that is important in the production of RNA is going to be RNA polymerase. So first we draw a circular RNA polymerase over here, right? So this is the RNA polymerase. Now what we do is we draw the first strand of DNA, okay, and then eventually we are going to draw the second strand, because we know DNA is double stranded, but on each strand of the DNA we have nucleotides that are present. So we have drawn these nucleotides right over here. Now comes the next strand over here, and what we see over here is, what is the RNA polymerase doing? It is separating the two strands of DNA, because RNA is going to be made up of just one strand, and for the production of the RNA only one of the two strands of the DNA is going to be required. So it is the job of the RNA polymerase to separate the two strands so that one of the two could be read easily, right? So that is exactly what it is doing over here. We draw nucleotides over here as well, rather nitrogen bases on the top part as well, right? Then what we do is we are going to label them, right? Once we label them, like the nitrogen bases like A, U, G, C, all these different letters we are going to label them with, right? In fact, for DNA it's not going to be uracil, it's going to be thymine, right? Please make a note of that. So we have labeled them. One thing we need to remember over here, whatever letters we use on the bottom strand, on the top strand the letters have to be complementary. Like for example, on the bottom strand if we have adenine, on the top strand just exactly opposite to it there has to be a thymine. So we need to be careful about that. Okay, now let's start drawing the mRNA molecule. So as the RNA polymerase is sliding along the DNA, it keeps on adding nucleotide after nucleotide, and we have the RNA molecule over here. We draw the nucleotides, the nitrogen bases in the RNA molecule as well, and again we need to be careful over here. Whatever nucleotides or whatever nitrogen bases we are adding in the RNA molecule, they also need to be complementary to the bottom strand, with one very careful detailing, that thymine is going to be replaced with uracil, right? So the correctors might cut marks for that mistake, so we need to be careful about that over here, right? Then what we show is the incoming nucleotides. Obviously the mRNA molecule is made up of nucleotides, and these nucleotides need to come from somewhere, so we show those incoming nucleotides, and now we start labeling the diagram. So obviously we have the DNA, the double strands of the DNA, we have the direction of transcription, the direction in which the RNA polymerase is moving, then we have the RNA polymerase itself, the RNA nucleotides that are incoming, right? So that was about the transcription. Now we label about translation and translocation. In the second part of this question, we need to draw the diagrams and label translation and translocation. So obviously it starts with the nucleus, because the mRNA, which is a very important ingredient in translation, comes out of the nucleus via the nuclear pores. So we draw the nucleus over here, right? We'll draw the nuclear pores, and from one of the pores we'll show the mRNA molecule moving out, coming into the cytoplasm. Once in the cytoplasm, we know it is attached by a ribosome, and the ribosome will start translating it. So we draw that same mRNA is going to be repeated over here. We draw it over here, and on top of that we are going to draw a ribosome, right? Now the ribosome will inside it have tRNA that are incoming. So we have a tRNA that has already attached. We'll draw one more next to it, right? Because the respective tRNAs bring their respective amino acids. The amino acids need to be next to each other to make a peptide bond between them. So that is exactly what we have done over here, and we'll show one more incoming tRNA with its respective amino acid. Now we start labeling it. So we have the initial codon, the first codon onto which the first tRNA is attached along with its anticodon. Then we have the amino acids that is present. We can label it only on one tRNA. We do not need to label it for each and every tRNA. Then we have the mRNA molecule that is coming out of the nucleus itself, right? The tRNA over here, the transfer RNA that is carrying the amino acid. Then we have the ribosomes on the second part over here, right? And finally the same mRNA but present in the cytoplasm. That is how you draw and label the diagram for transcription and translation. In fact, translation and translocation.